Hello grade 11 students. Today I am going to discuss chapter 1 of your science textbook. This is a biology lesson, living tissues. In grade 10 you have learned many different lessons and there were many related to biology. And you have a fundamental idea about living organisms, structure of the body of living organisms. And under that, you all have discussed so far about microorganisms, plants and animals. And also, you have a good idea about the characteristics of living organisms. And in that, you know what organizational levels are. All the organisms are made up of the simple unit cell. From there onwards, the complexity increases. The cells combine and form tissues, tissues combine and form organs, organs together make up the system, and many systems together make up the body of a multicellular organism. And in this chapter, we are going to discuss the second level of organization, that is the tissues. So first, we will see the contents of this lesson. In this, we will be discussing plant tissues and animal tissues. You already know, students, plants and animals have different types of body structure and also they perform different functions. Therefore, the basic structural unit, the cell itself is different. We had the plant cell and the animal cell. Similarly, we have the two main different types of tissues, the plant tissues and animal tissues. So, we will start off the lesson with an activity to observe how tissues will look like in living materials. So, the first activity is to see how plant tissues will look like. So, study about plant tissues. This is a simple activity where we use 3 different parts of different plants. And the first one, we use a beetle leaf. And from this beetle leaf, you all know on a leaf, there is an upper epidermis, the upper part of the leaf, and the lower epidermis, the below part of the leaf. And in the lower epidermis, there are more stomata, the opening or the pores present on the epidermis so that air can be exchanged. Air can go into the leaf and come out of the leaf. So, those stomata are largely present in the lower epidermis. So, here we will be observing the lower epidermis of the beetle leaf. Then the second plant part is going to be a potato tube. So, this is a potato tube. This is the underground food storage stem of this potato plant, a potato tube. Then we have the third specimen, that is a stem. You all know this is a balsam plant. And that has a very thin stem and a light colored stem so that it will be easy to observe the cells and the tissues present in it. So therefore, we will be using a cross section of the stem. Cross section of the stem. So the activity is this. We will be preparing temporary slides. We have to take a thin peel from this beetle leaf. There are two ways of preparing that thin peel. You can either use a very sharp blade and cut thin peels, or you can just peel the leaf and get a very thin layer from that. So we can get a thin peel of the beetle leaf. Similarly, we have to cut a thin peel of the potato tube, a thin layer of the potato tube. You can cut the potato into half or into 4 pieces. Then from the exposed surface, we can cut out very thin layer so that we will be able to observe the minimum number of layers of cell. And the third one again, we have to cut a cross section of the stem. So from the middle part of the stem, we have to cut a very thin cross section. Now each one has to be done separately. We will start off with the beetle leaf, cut a thin peel or peel out a thin peel of the lower epidermis of the beetle leaf. We will take many peels as possible, put it into a watch glass containing water. Then we need a clean glass slide. On that we will put a drop of water. Using the paintbrush, transfer the thinnest peel onto the glass slide and then cover it with a cover slip without tapping any air bubbles. Then the slide will be ready for observation. You can observe the slide through the light microscope, and in that also we will observe through the low power first and then we will move on to a higher power. The same procedure will be repeated with the potato tuber and the balsam plant. So let me summarize the method for you all. Prepare temporary slides using above plant materials. So you can either peel or cut off a very thin layer from the plant parts. As I told you, the beetle leaf. Cut a very thin layer of the plant part, that again potato and the balsam stem. Observe them under microscope. And then the main reason of this activity is try to identify tissues formed by the cells. So that is our aim. We have to observe how the tissue will look like or how the cells will be arranged in this tissue. A very simple activity, and you all are familiar with the procedure of preparing slides. So let us do this activity in the lab. Okay students, so we are going to start off the lesson with the first activity. What are we going to do now? We are going to observe the cells present in different types of plant parts. So for that, I have a beetle leaf. We will be observing the lower epidermis of the beetle leaf. Then we will need to observe a very thin piece of the potato tuber to observe the cells present in it. And the third specimen, it is a balsam plant. It has a very thin stem, so we will be observing the cross section of this stem. I will start off the activity by preparing a temporary slide of the beetle leaf lower epidermis tissue. So for that, we will have to take the beetle leaf and you all know this is the upper epidermis and this is the lower epidermis. So here we use the lower epidermis because there are more stomata present in the lower epidermis. We can take thin peels by cutting very thin pieces. So I can do that as well. Here we will have to cut very thin peels first. For that we have to use a very sharp blade so that you can get it as thin as possible. And once you cut off small thin peels, you have to put it into a watch glass containing water. It can be any glass container containing water. Then I have the brush to transfer the specimen onto the glass slide and the cover slip to cover the slide. So first I will try to get the thin peels. Like this we can cut thin peels. We have to make sure it has to be a thin layer and also it has to be a whitish color layer. If it is too thick or green in color, then of course there can be many layers of cells that will not be very clear to you. We can do it this way or there is another method. Here what I have done is I have taken a piece of the leaf and if you tear it like this, also you can get very thin peels. And from that you can see it's a very thin layer. I can cut off the thin peel. And that also can be used as the tissue specimen. Then what do? We have to take a clean glass slide. I have one here now. And then using a paintbrush, I will transfer the thinnest specimen from water. Before that, I will put a drop of water on the glass slide. You all know why? So that the tissue doesn't dry off. It will be fresh and we will be able to observe the parts of it or the cells present more clearly. And then I'll transfer the thinnest specimen. Since it's a slightly large specimen, I will put another drop of water and on that I will be placing a cover slip. Have to take out the cover slip. It's a very thin cover slip. So what do we have to consider here? We should make sure that there are no air bubbles trapped inside. So for that, I will place the cover slip like this and drop it onto the specimen. Then of course, there will be no air bubbles trapped inside. Now the temporary slide is ready for observation. So now we have to check whether the microscope is ready. You can see the light is on, so there is enough light falling onto the stage. You can observe the specimen clearly. Then I will make sure that we select the low power and also I will place the slide prepared by me on the stage and adjust it so that the specimen comes into the correct position. Okay, now it's all set. Then we will observe the tissues that are present in the lower epidermis of the beetle leaf. Okay, so first I will adjust the coarse adjustment knob and see. Now students, you can see the cells present in the lower epidermis. Can you all see dark color like oval shape, dark spaces? You all know those are the stomata. And around that you can see two bean shaped cells. Those are the guard cells and also the rest of the cells are different in shape and also they are green in color. So these are the different types of cells present in the lower epidermis. So students, that was the observation through the low power. You all saw the stomata, the guard cells and the different epidermal cells. Now to observe it more clearly, I will change the power to a higher power. Now I am changing it to a higher power and now we will observe the specimen again. If I do a little bit of adjustment, you can now see it clearly. The cells, the shape of the cells are more clear to you. You can see the greenish color cells and also you can see the different shapes. The shape of the epidermal cell as well as the shape of the guard cell. In the middle of the guard cells, you can see the stomata clearly. So students, you have observed all the different types of cells that are present in the lower epidermis and you can identify the different known cells. Okay, so that is the first plant part that we are going to observe. Then now I'll move on to the second plant part, that is a potato tube. So now I have cut a small piece of the potato tuber. This is the part that we are going to observe. So from this part again, I will have to peel out or cut out very thin layers. So here you will have to cut very thin layers. You know the reason why we need to cut very thin layers. Thin layers will give you less number of layers of cells so that it will be very easy for you to observe it. And as we cut off the layers, we have to put it into a watch glass containing water like we did before. I'll cut out few more pieces so that we can select the thinnest piece from the pieces that we have cut out. Okay, now I have cut a few pieces there and I have the specimens from the potato tuber ready. Then I need to take a clean glass slide and onto that I have to put a drop of water. You all are very familiar with preparing temporary slides. From smaller grades you have done this. So therefore I am not going into detail about the preparation. And from this I select the thinnest piece and there is enough water. Onto that I am going to place a cover slip that is also glass. And what is the consideration here? We have to make sure that there are no air bubbles trapped inside. So for that we hold the cover slip slant like this and place it on the specimen. Now I have prepared the temporary slide containing a thin layer of potato tube. Now I will have to remove the slide that I placed earlier. This is the beetle leaf. I place the new slide I prepared. Okay, again I have to make sure the specimen is in the correct position. That's correct. And also I have to adjust the lens to the low power. Low power is ready. Now I have to observe it. So first I will observe it through the low power and with a little bit of adjustment you can see, students, clearly the shape of the cells. You can see almost all the cells are spherical in shape and they are all similar in size. That is the observation you can get from this slide. All the cells of the tissue are similar. So that is not clear enough. Then what do? I will change it to the higher power so that we can get a more clear observation. Now with this higher power, when I do a little bit of adjustment, now you can see more clearly the spherical shaped cells, and they are almost the same size, and there are spaces in between the cells, and these cells all look very similar to each other. So students, you can get the observation clearly how the cells of the potato tuber look like. It was different from the first slide we prepared, that is the lower epidermis of the beetle leaf. Now again, I will be using the third type of plant part, that is a stem. And we will see how that will differ from these first two slides. So I will remove this slide. I am going to use another slide. I have to prepare it first. So here you can see, students, this is a balsam plant. Now we usually take this balsam plant for the evenness and the thin skin, and also because of the color you can observe the cells clearly. So here you can see this part is a little darker in color, this part is slightly light in color. I will be cutting very thin cross sections from the darker part of the stem. So first I will have to cut out small specimens. They have to be very thin. And again, to make sure that we get very thin pieces, I will cut more than one layers. Here you can see I am getting very thin layers. Those are the cross sections of the balsam plant stem. I will cut one more. Right. So now here you can see it's all colored. So here itself you can observe slightly the different regions. When we observe this through the microscope, that will be very, very clear to you. So now I am going to make the slide again a clean glass slide. I will put a drop of water. I might need a larger drop if I am using a very thin layer. I will take the thin layer and transfer it onto the glass slide. So here the specimen is there and there is enough water. So I have to put a cover slip onto that. So for that, I have to make sure that there are no air bubbles trapped inside. I hold it slant and place it on the specimen. Now the temporary slide mount of the cross section of the balsam plant stem is ready. I will first adjust the power to low power, then place the glass slide, that is the temporary slide, and make sure that the specimen is in the correct position with enough light falling on it, and now it's ready to observe. If I adjust it and observe, you can see the cells are reddish brown in color because of the color of the stem. So you can identify the different cells. Here you can see in the middle part of the stem there are very tiny cells. Whereas when you go towards the outside, the size of the cell increases, and towards the periphery, you can see oval-shaped dark color regions. Those are the transport tissue, that is the vascular bundle containing the different types of cells. We will discuss that later. For now, you can observe the difference in the size and the shape of the cell. So, you have observed 3 different parts from 3 different plants, and you have seen that there are different types of cells which exist together to form the tissues of these parts. And when you saw, the beetle leaf had green colored cells, whereas the potato tuber had identical spherical shaped cells which looked similar. And the stem of a balsam plant had different types of cells present in it. But there were many similar cells. That is what we call as the tissue. So, from all these observations, you all can understand that different cells are there. They have shapes and sizes, and similar cells together forms a tissue. Okay, students. Now you were able to observe the different types of cells present in the different tissues that were obtained from 3 different plant parts. First, we will discuss the lower epidermis of the beetle leaf. What did you all see? I am sure you were able to observe dark colored lines like these, which are the stomata, and around that you were able to see this type of kind of a bean shaped cells, that is the guard cells. You are familiar with that. And around the guard cell you were able to see something like this, an irregular shaped cell. So here this part is the stomata, and these cells are the guard cells, and this is the epidermal cell. So this is from the lower epidermis of beetle leaf. So when you observe the lower epidermis, you were able to identify different shaped cells, especially the guard cell and the epidermal cells were different to each other. And you saw that there was a particular arrangement of similar cells. This was different from the cells that you observed in the second specimen. That is a thin section of the potato tuber. I am sure you were able to observe cells like this. Like this was visible to you in the potato tuber. This is different from the first type of cells. So here you can see there are different types of cells and tissues present in plant parts. Then we looked at the third specimen, that is the cross section of a balsam stem. In that you would have seen it is a circular shape like this. Students, I am only drawing a rough sketch of what you would have observed through the microscope. If you look using the very high power, you will be able to observe these even more clearly. But from what you saw, the observations that you got, I am drawing a rough sketch. In this cross section, you would have seen the darker color layer towards the periphery of the stem. So, you saw the dark layer towards the periphery, and you saw towards the middle there were different types of cells, and also you would have been able to observe small structures like this. And in that structure also you would have been able to differentiate the three regions when we observe through the high power. So, in this, these are different types of cells. Then these are the transport tissue or the vascular bundle that is present for transport. Vascular bundle. And the cells present in the vascular bundle would have been different to the cells present in the middle, and that differs slightly from the cells in the other regions. So, from all these observations, what can you conclude? I am sure you can conclude that there are different types of cells present in a plant, and also these different types of cells are arranged together to produce or to have different types of tissues. So, those are the conclusions. We can say there are different types of tissues present in different parts of the plant. And we can say similar cells arrange together and form tissues. And here we can say similar cells exist together to form the tissues. So, from this observation and what you have learned in your previous grade, we can say that tissues are a group of cells that have a common origin. They are produced from the same type of cells. So, with a common origin and are adapted or modified to perform various functions. So, when we say that, now this, the lower epidermis, the cells also had green color pigments. Why? The leaf does photosynthesis. So, these cells are mainly adapted for photosynthesis. Whereas the potato tuber, it stores starch. So, this is for storage of food. And this cross section of the stem has many functions. Especially the transport of substances is done by vascular tissue. So, therefore, tissues are a group of cells with a common origin that are adapted to perform a specific function. So, we can put that into words, a group of cells with a common origin that has been modified to perform particular functions in the body is known as a tissue. So, in this you have to remember these terms, common origin, and they perform particular functions. So, because the plants and animals are different from each other, there are two main types of tissues. So the first one is the plant tissue that we observe now, and second one is the animal tissue. Now we will go into detail about plant tissues and how they are classified further. For that again, we will do an activity. So for this activity, we need only one material, that is a plant root. For this, we can use any one of the plant roots, that is a stilt root of banyan. We can use a stilt root of banyan. You know what a stilt root is? The roots that are formed from the branches to balance and bear the weight of the huge plant, the huge tree actually. Or we can use the prop root of pandanus, or we can use the prop root of rampe. I will be using a prop root of rampe, and this is what we are going to do. We are going to observe the external view of the growing tips of above roots. You all know, students, the shoot grows, the plant height increases, and the root grows so that it can grow and hold on to the soil as well as absorb enough water and minerals. So what we are going to do is we are going to observe the growing tips of the root. Always, if this is the main root, this part will grow further, and from that you can see many side roots as well. And here we will be observing at the base of the stem there are prop roots. So we are going to observe the prop root of rampe, and then we can observe it more clearly if we use a hand lens. Since this is a very simple activity, we will now do it in the lab. From the first activity, you know how tissues look like. And you know what tissues are? A group of cells that have the common origin and that are adapted to perform a certain function. So now we will be looking at a plant part. For that, I will be using a rampe root. And we will use this to see how the external view is different based on the type of tissues that are present in the plant. So here you can see this is a rampe plant. There is a very large plant here out of the rampe plant, so that you can see the parts clearly. And in this you can see all these stilt roots. The main root is there in the middle, and there are the stilt roots that start from the base of the plant. That is to support the plant to stay erect in the soil, which is a muddy, loose soil. So we will be observing the stilt root. Here you can see there are different stilt roots, and this is a clear root that you can observe. And here you can see the shape. It's broader and almost the same diameter, but towards the end of the tip of the root, you can see it becomes a little more thinner and gets pointed at the end. And you can observe a color change. Here it is a darker color, almost like a brownish color, but when you go towards the tip of the root, you can see the color becomes lighter and green, and this is a light yellowish green in color. And here also you can observe the different roots with the different color shape. What is the reason for this color change? Here there are cells inside that are mature. So because of that, they are more stronger and also darker in color. But here there are cells which are dividing and somewhat immature. That is why you see the lighter color. So from this observation, you can understand, depending on the type of cells, the part of the plant shows different color and structure. So then only the cells can do their function, or the cells that make up the tissue can carry out their relevant function. That is the observation of this activity. You were able to observe the root of Rampe plant, which is the prop root in the lab. You saw the difference in parts of that root. You saw that the tip of the root was soft and also light in color, whereas the mature part was dark in color and also rough. So from that, you know that different parts, although it's the root, different parts of the root have different nature because of the type of tissue that is present inside. Now, if we take the root, the tip of the root is to grow. The adaptation is to grow long, whereas the mature part has been well developed. So, that is the observation that we got from this activity. So, if we say observations, the tip is soft and light in color, and the mature part is rough and dark in color. So these were the observations. From that, what can we conclude? There are different types of tissues in the growing part and different types of tissues in the mature part. So I can say the tissues present in the growing part of a root tip is different from the tissues present in the mature part. So from this, you know that there are different tissues in the part that grows continuously and different tissues in the mature part. Now let us continue this activity to observe the longitudinal section of the prop root of Rampe, so that you can observe the types of tissues even more clearly. Activity. microscopic view of longitudinal section of a root. So, to observe the longitudinal section, we will use the same root, that is the prop root of Rampe, what we used in the previous activity. So, here I can say prop root of Rampe, or you can also use the roots of banyan or else even the root of pandanus. Any root can be used. Since I use the prop root of Rampe, I am going to use the same root here. So then what we will do is we will choose the tip of the root. So it will be a part like this, the tip of the root with the root hairs and all those here. From this we will cut a small section, and from that section, that tip of the root, we will have to cut very thin longitudinal layers. So longitudinal is going to be this way, you cut it into half. Then we need to cut very thin layers of the longitudinal section. And since we are going to observe it immediately, and this is anyway a somewhat dry part of the tissue, we might not need to put it into water. We can immediately place it on a clean glass slide and observe it under the light microscope. But if you are going to use it after some time, it's better to have it in water as we normally do. So then I will need a glass slide. And once we have the specimen ready, we have to observe it under the light microscope. So this is a very simple activity where we take the longitudinal section of the prop root of Rampe and observe it under the light microscope to see whether there are different types of cells present. You have to observe their color, the size and the arrangement. So let me move on to the method. First we observe the nature of the prop root of a Rampe plant. Now we will get a longitudinal section of the part of the root and then observe it in the microscope. For that, I will select one of the roots which shows a color difference. So for that, I can select this root and I'll be cutting a small piece from that root. Okay, so I won't be needing the plant anymore. Here I have the part of the root. Here also you can observe the slight difference in color. Then we will have to cut a longitudinal section. So I have to cut it through this root. For this, I have to use a very sharp blade so that it does not damage the cells. And from this, again, I will need to take a very thin layer. And we have to make sure that we get the long, thin layer starting from the point till we get to the tip. So I have to be careful when I cut the longitudinal section. Again, I will take another. I will make it a bit shorter and take another section. Now I have got a thin layer. So the next step, you all already know, we have to take a clean glass slide, which I have it here. And on that, I will use the blade to place the section. So I have the part of the root here. This is a longitudinal section, and since it's a larger part, I am not going to use a cover slip. So, but I have to make sure that the specimen does not come in contact with the lens of the microscope. So now I am going to observe it under low power. I'll start from the base of the root, that is the part where we cut it off from the root. So if we look at the cells there, you can see, students, the cells are in the middle. You can see many layers of cells, and the cells are very much identical or similar in shape and size. And we will see how the size changes when we go from the base to the tip of the root, and also towards the outer line of or the periphery of the root. You can see that there is a darker color region. Now I will move the specimen towards the tip so you can observe it clearly. Now observe the size and the color change in the cells present. Can you all see that the color is becoming darker and the cells are becoming smaller? So students, in this you can observe that towards the base, that is the part where it's connected to the root, the cells were larger and lighter in color. And when we go towards the tip of the root, the cells become darker and smaller in size. So with that observation, you can understand even in one part of the plant, there are different types of tissues. The tissues that are dark and small in size are the meristematic tissue. And the tissues which are more larger and which have a distinct shape and lighter color are the permanent tissues. I am sure you were able to observe the different types of cells or the tissues present in the longitudinal section of a root. So here you would have seen it was very much similar to this diagram. And here there are two parts marked in the diagram, the section AA and section BB. As you can see, this part is the root cap. It looked very dark in color and the cells were very tiny. You were able to observe that. Then what did you see in this section AA? Were the cells small or large? You were able to observe very tiny cells which were very similar to each other. So in this observation, when you observe the part AA, there were very small cells, almost like very tiny squares like this. And you all were able to see a distinct nucleus in the middle. A dark color, almost black color dots in the middle. That was prominently visible to you. Then when we look through the section BB, when I moved the slide while observing through the microscope, you were able to see the cells were more large. The cells were large, and you would have been able to see that almost the cells were elongated like that. And there also you were able to see a nucleus in each cell. So if you observed carefully, you would have seen that the cells towards the tip, that is the section AA, was smaller, whereas the cells that were towards the mature part, they were more larger. And towards the periphery of the root, the cells were more darker. So from this, we can conclude that there are two main different types of tissues present in a plant. In the growing part of the plant, the cells or the tissue present is adapted to divide and make sure that the root grows. And we will be discussing that tissue next, and this is called the meristematic tissue. And towards the mature parts, there are more adapted and modified cells which do not divide like the meristematic tissue. Then we call them the permanent tissue. So these are the two main types of tissues based on the ability to divide. So let us classify the plant tissues first. Plant tissues can be categorized using different criteria. There are many criteria that can be used. Plant tissues can be divided into two groups according to the ability of cell division. That is what we observed a little while ago. So under that, there are two types. You know the names by now. Meristematic tissue and the second one is permanent tissue. So meristematic tissues have the ability to divide all the time, whereas permanent tissues do not divide, but they are differentiated and adapted to perform specific function. Now we will discuss the features and the uses or functions of meristematic tissues. Meristematic tissues. The tissues with cells that divide actively by mitosis to produce new cells are called meristematic tissues. So here you have to remember, students, tissues, they divide actively by mitosis. What is the significance of mitosis? You have studied this in grade 10. There are two types of cell division, and out of those two, in mitosis, identical cells are produced from the mother cell. If the mother cell is 2N, that is diploid, the daughter cells that are formed are also diploid cells. So that is useful in growth. Here again, the meristematic tissue is going to divide and make the root or the shoot to grow. Therefore, it has to divide by mitosis. And they say actively divide. So it's like dividing all the time. And because of that, these cells are not differentiated. So as we saw in the longitudinal section, all these cells look very identical, small, and they don't have different specific shape or features. That is what we mean by not differentiated. The basic type of cells are present. The growth of plants take place due to activity of meristematic tissue. So here these tissues are important in growth of plants. So, shoot apex contains meristematic tissues, and when they divide, the height of the plant increases. Height of the plant increases. And there is meristematic tissue in the root apex. This leads to the elongation and growth of roots. So here I can say elongation and growth of roots. So this is the main function of meristematic tissue. And there is another function, increase in the diameter of the stem. That is also due to the division of meristematic tissue. So the third one is increase in diameter of the stem. So these are the different types of meristematic tissues, and their main function is to divide and give rise to growth. So based on that, meristematic tissues have certain features. Let us now discuss that. Features of meristematic tissues. These are all living tissues. Why? They divide all the time. So because of that, they have to be living tissues. So that is the first feature. Cells are living. And because they divide all the time, the cells are small in size. Cells are small in size. And you know, students, during the process of cell division, what divides first? It's the nucleus. So because of that, meristematic tissues, the cells contain a distinct nucleus. A nucleus that is very prominent. So cells contain a distinct nucleus. And also, because they are dividing all the time, the cells are not differentiated. Cells are not differentiated and for the cell division, these cells need a lot of energy. Because of that, there is a large number of mitochondria present in these cells. Large number of mitochondria are present in the cells. And also, normally there can be space between the cells, that is intercellular space. But here the intercellular space may be absent or not prominent. So the next one is intercellular cellular space may be absent or not prominent. So, in addition to these features, there are two more features of the meristematic tissue. Because the cells are dividing, their nucleus is distinct and therefore the central vacuole is not present. So, that is, these cells do not have a large central vacuole, but there can be small vacuoles present. So, that is the next feature. Large central vacuole is absent. Small vacuoles may be present. And because these cells mainly divide, they do not do photosynthesis. Therefore, although they are plant tissues, they do not have chloroplast. Chloroplast is absent. So, you can see, students, all the features are related to the function of the meristematic tissue, and they divide and help the plant to grow in different ways. So, now let us see where they are present in a plant and how they help the plant to grow. Meristematic tissues are present in specific locations of the plant. They are of three types. The first one is the apical meristem. Apical meristematic tissue or apical meristem. The second one is lateral meristem. And the third one is intercalary meristem. Now, we will see where these meristems are present and what they do in the plant. Here you can see apical meristem, intercalary meristem and the lateral meristems. Look at the diagram first. This part is the shoot apex of the plant. The part that is on the the tip of the plant which grows and increases the height. So, there we have the apical meristem because it's found on the apex. Similarly, it is present in the shoot apex as well. So, here it is present. You can see apical meristems are found in shoot apex, root apex and also the axillary buds. So, growth of the plant to increase the height, that is the shoot apex, growth of the root to increase the length and also axillary buds are the places in the plant from where the branches, the leaves, flowers, all that are growing. So, in those axillary buds also we get apical meristems. And the function of this apical meristem, plant increases its height due to the activity of this tissue. So, this is the position of apical meristem. Then we have the intercalary meristem. This is mainly observed in the plants of the grass family. You would have seen, students, if we look at the stem of the grass, it will be like this, like small joints are present in the middle. So, those parts where it looks as if the stem is joined together is the node. This is one node, this is another node. And in between the two nodes is the part that is called the internode. And this intercalary meristem is present in these parts. So, here you can say intercalary meristems are found at nodes. The length of internode increases due to the activity of the above tissue. So, the plant increases this height. Each node increases in length. So, the plant grows. They are found in plants of grass family. So, here the length of internode increases. So, you can see this part is the intercalary meristem, and when that grows, the cells divide, the length of the node increases. So, overall there will be an increase in the height of the plant. Then we have the third type, the lateral meristem. Here you can see this is the longitudinal axis of the plant, and the lateral meristems, these two are the lateral meristems. They are parallel to the longitudinal axis. So, if this is the axis, the lateral meristem is present around it. That is what is shown here. This is the axis and the lateral meristem is around that. So, if you look at the longitudinal section, it looks parallel to the longitudinal axis. This makes the plant to grow sideways. That is to increase the diameter of the plant. And these types of meristems or meristematic tissue are present in dicot plants only, where you see the secondary growth or what we call as the woody plant, in which the wood is formed. The stem becomes broader and becomes woody. So, if we look at the lateral meristems are present laterally in the stem and roots of plant. They are found parallel to the longitudinal axis. The diameter of the plant increases due to the activity of this tissue. And this is given a special name. Cambium tissue found in dicots is a lateral meristematic tissue. If you can remember, students, we observed the cross section of the balsam plant. And there you can remember observing structures like this, which I said is the vascular bundle. In the vascular bundle, there is a region in the middle where the cells are different. And that is the cambium. And this is the vascular bundle. So, this is there to divide and increase the diameter of the plant stem. And that is also in dicot plant only. So, that is what you need to know about meristematic tissue. Now, let us go on to the next type of tissue, that is the permanent tissues. To observe and learn the permanent tissues, let us do this activity. Identify different types of tissues of plant stem. So, for that we will need a thin stem, where you can take the cross section easily and observe the different types of tissues present there. For that, I will be using a pumpkin stem. And from this pumpkin stem, we will be cutting out very thin cross sections using a blade, and we will transfer those thin layers into a watch glass containing water, and then we need a clean glass slide. And onto this clean glass slide, as you already know, put a drop of water and transfer a cross section using a paintbrush, and then cover it with a cover slip without trapping any air bubbles. And when the slide is ready, we observe it under the light microscope. And that also under low power first and then under a higher power. Let us look at the method again. Observe the cross section of above plant stem using a microscope. Why are we doing this activity? To identify the different tissue types present in them. So, let us do this simple activity in the lab now. We are going to do this activity to observe the different types of tissues present in a cross section of a plant stem. So, for that, we can either use a pumpkin stem or a tridac's stem. Here I have a pumpkin stem. It's only a part of the stem. From this, I will be cutting very thin cross sections to observe using the temporary slide. So, you know the procedure. We have to cut very thin layers of the cross section, and also as I cut the cross sections, I have to put it into water so that it doesn't dry off quickly. So, now I have taken 2 cross sections. I will take a few more so that we can select the thinnest one. Right. So, I have a few thin cross sections of the pumpkin stem. So, what do I do next? I need to take a clean glass slide, and onto that I put a drop of water, 1 or more drops, so that the specimen will not dry off. Then from the layers I have cut out, I will select the thinnest one and place it here. There is enough water, and on that I have to place a cover slip. I will place the cover slip onto that slide. And what do I have to consider? When placing the cover slip, I have to make sure that air bubbles are not trapped inside. So, now the temporary slide is ready for observation. I will place it on the stage as we normally do, and make sure that the specimen is in correct place and there is enough light falling on the specimen. Okay. So, first, what is the power that we have to use? It is the low power. So, through the low power, we can observe the full area and see how the cells look like. So, here when I do a little bit of adjustment, you can see, students, there are different layers of cells. In the middle, you can see different shape and different size, whereas towards the periphery, you can see different size and shape. And also you can see the darker regions, which are the vascular bundles or the transport tissue, which contains the xylem and phloem, which we will be discussing later. So this is under the low power. You have already observed this before. What we are going to do now is we are going to observe a small region across under high power to differentiate the cells more clearly. So I will change to high power. Then I do the slight adjustment. Here you can observe the size and the shape of the cells. Towards the middle, you can see there are very tiny cells. But when you go towards the outer circumference of the stem, you can see the size of the cells increasing and the shape is more prominent. You can see polygonal shaped cells. And also in between, you can see the darker regions. You can see that clearly, students, the darker regions, that is the xylem and the phloem. In between the xylem and the phloem, where you have the middle line, those cells look very small and you can identify that region. So that is the meristematic tissue. So the rest of the cells that you are able to identify are permanent tissues, and the cells in the middle, which is the cambium, contains the meristematic tissue. So from this observation, students, we have identified the two main types of cells based on cell division. The cells that divide all the time, or the main purpose of the cell is to divide, are the meristematic tissue. And that was the cambium layer you saw between the dark regions. That is the xylem and the phloem. And the rest of the cells that you saw in different shape and size, especially the polygonal shaped cells, they come under the permanent tissue category. So with this observation, you all now have an understanding about the two types of tissues, meristematic and permanent tissue. Okay, students, now you observed the cross section of a pumpkin stem. And I am sure you would have been able to identify cells similar to this. So you would have observed this under the light microscope. Here you can see in the middle part of the stem, there are different types of cells. Similar type of cells which are usually small in size. And then towards the periphery, the outermost layer looks darker in color, and slightly below the outermost layer, you would have been able to observe these regions. These are the vascular bundles. We have discussed that before as well. This has the transport tissue. And if we look at this tissue, a part of it under high power, you would have been able to see that there are different types of tissues like this, various regions. So now you know that this is the permanent tissue that has been differentiated. That is why they have different shapes and different sizes, as we can see in the diagram. So these are differentiated tissues and they perform specific functions. So they are different from the meristematic tissue that we discussed before. And under these permanent tissues also, we have different types. We will be discussing it after this. So here from this observation, you can say there are permanent tissues in the plant with differentiated cells. That is why the shape and size is different. The shape and size of cells are different. So from this observation, let us discuss the different types of permanent tissues. A specialized tissue that lost its ability to divide and perform a particular function is known as a permanent tissue. So here you have to remember these terms: specialized tissue, lost its ability to divide, and it performs a particular function. So then we call those types of tissues as permanent tissues. And according to the nature of permanent tissue, that is the types of cells present in the permanent tissue, we divide it into two groups. One is called the simple permanent tissue, this tissue has only one type of cells. Only one type of cells. And the second type is complex permanent tissue. This has more than one type of cells. So let us first discuss the simple permanent tissues present in a plant. Simple permanent tissues, the tissue is composed of similar cells. According to the shape of the cell and the nature of the cell wall, three types of simple permanent tissues are there. And here you have to remember, students, the shape of the cell and the nature of the cell wall. The three types are Prenchyma, Collenchyma, and Sclerenchyma. Now let us see how the shape of the cell is different in these three types of tissues and also the nature of the cell wall. This diagram shows you the Prenchyma tissue, Collenchyma tissue and the Sclerenchyma tissue. Here you can see there is a thin cell wall and the cells are spherical in shape. Whereas the Collenchyma, it has polygonal shaped cells. This is a polygonal shaped cell. And also here you can see the cell walls are very thick at the corners. So that is a feature of Collenchyma. And if we look at the Sclerenchyma, you can see again they are polygonal shaped cells. They have a thick cell wall because there is a substance called lignin in the cellulose cell wall. We will discuss those in detail, but you can see the cell wall is evenly thick the thickness is even. And inside the cell, the middle part is like a space, which is called the lumen. So these are the three types of simple permanent tissue. As you see the type of the cell, you should be able to identify the type of tissue. Now let us discuss the nature of parenchyma tissue. Parenchyma, the tissue that forms the soft parts of the plant body, is the parenchyma tissue. So here it's the soft parts of the plant body. This is the most abundant tissue found in the plant. Again, as I said, you can see the cross section of the parenchyma tissue. The size and the shape is different, and also the nature of the cell wall. Those are the things that you have to observe here mainly. So if you look at the cells, these are spherical in shape. We call them as isodiametric cells or spherical shaped cells. And here you can see there is a thin cell wall. So thin primary cellulose cell wall, and inside that you can see the large central vacuole. Because of the large central vacuole, here you can see, students, there is only a small area of cytoplasm, and in that cytoplasm the nucleus is present towards the periphery. So in the cross section, parenchyma looks like this. If we take the longitudinal section, you can see these are elongated cells, and you can see the nucleus and the large central vacuole. That is the structure that you can see. And also in between cells you can see the space. That is the intercellular space. So these are the features of the parenchyma tissue. Let me summarize all these features for you. Features of parenchyma. These are simple permanent tissues and they are made up of living cells. And the shape of cell is isodiametric or spherical shape. And they have a thin cellulose cell wall, large central vacuole is present, due to the large central vacuole, the cytoplasm is towards the periphery. And also the nucleus is pushed to the periphery. And you saw that there is intercellular space present. Intercellular space. So students, these are the features of parenchyma tissue. Now that you have understood the features of parenchyma tissue, let us observe this using a permanent slide in the lab. Now we are going to observe the permanent slide of a parenchyma tissue. You know the features of parenchyma? What are they? The cells are isodiametric. That means they are spherical in shape. And also they have prominent intercellular spaces. And also they are living cells. So to observe this, I will use a permanent slide. Okay. I have this permanent slide of the parenchyma tissue, which I will place it on the stage. I make sure the light is on, there is enough light, and we have to move the slide to adjust. Okay, the specimen is in place on the stage. So first I will observe it through low power. Already the low power lens has been selected. Let us observe the parenchyma tissue. Now you can see, students, the parenchyma tissue, you can see the isodiametric shape, and also the cells are all similar to each other. We can actually say they are identical to each other. And you can see the intercellular spaces. So those are the features that you can see under higher power. So students, these are the observations that you can get by observing these permanent slides. Locations of parenchyma tissues: cortex and pit of plant stem, pit and cortex of roots. Now we have observed the cross section of a plant stem. In a cross section, if I put it in a diagrammatic form, this is the epidermis. That's the outermost layer of the cross section. And here you will have seen the vascular bundles present like this, whether it's a stem or a root. And in this, the cells that are present, or the part of the plant stem in the middle, is known as the pith. And this space, which has cells in between these vascular bundle, is known as the cortex. So these are the two places in both the plant stem and the root where you get parenchyma tissue. Fleshy parts of fruits. If we take fruits like papaya, banana, these are the fleshy parts which are edible. In that, the main type of tissue is parenchyma, and that stores the food. Then we have the seeds, the endosperm of seeds. If you take green gram, inside the seed there is the starch storage, or mainly the food storage. That storage is what we call as the endosperm, and this is made up of parenchyma tissue. Then we look at the leaves. If I just show it diagrammatically, if this is the cross section of a leaf, in these leaves you will see cells like this. Long elongated cells with a lot of chlorophyll in it. This is known as palisade parenchyma. Under that, you will see irregular shaped cells like this that also contains, this is just a diagrammatic representation, students. This is known as the spongy parenchyma. Both of them together is known as the mesophyll cells. And this is a type of parenchyma tissue. Now that you know where parenchyma is present in the plant, we will discuss the functions of parenchyma tissue. Functions of parenchyma. First one is photosynthesis. Palisade and spongy mesophills. What I explained to you before. In plant leaves contain chlorophyll within chloroplast. Chloroplast is the organelle that contains the chlorophyll pigment, and photosynthesis takes place within these chloroplasts. So you have to remember the two names, palisade and spongy mesophyll cells. The second one is food storage. Food is stored in some parenchyma tissues and they are called as storage tissues. We observe the potato tuber. Their food is mainly starch is stored inside this parenchyma tissue. Carrot and sweet potato roots. Pepper and banana fruits. So different parts of the plant where food is stored, it is stored within parenchyma storage tissue. Storage of water, especially xerophytic plants store water in parenchyma tissues. You know there are different geographical locations where there is very less water and the temperature is very high. Normally it is a very warm environment. Then the plants have to prevent loss of water and they need to have methods of storing water. This is seen in plants like aloe leaves, bryophyllum and cactus, and these plants are known as xerophytic plants. Providing support. Herbaceous plants like balsam absorb water into vacuoles of the parenchyma cells, thereby cells become turgid and provide mechanical support to the plant. Herbaceous plants are the very small plants. Here they don't have wood in their stem. The stem is made up of very thin, soft tissue. So because of that, water is retained within the vacuole, so that turgid means it becomes bloated up and it provides a turgid pressure. So due to this pressure, the stem of the plant is kept erect. So there it provides support to herbaceous plants. So students, these are the functions of the parenchyma tissue. This is the first type of simple permanent tissue present in plants. Now let us move on to the second type of simple permanent tissue. Collenchyma. Collenchyma tissue provides mechanical strength and support to the plant body. So here it is mechanical strength and support to the plant body. And these are modified parenchyma cells. Now you saw this diagram before, a cross section of a collenchyma tissue. What are the features you can notice in the diagram? Here you can see the cells are polygonal in shape, which is different from the parenchyma tissue. And also you can see this cellulose thickening at the corners of the cell wall. The cell walls are not even. The corners are more thick because of the cellulose deposit. That is a very distinct feature of the collenchyma tissue. But these are also living cells. Therefore, they contain the nucleus, cytoplasm and the central vacuole. If we look at the longitudinal section, you can see these are elongated cells. And here you can see the corners of the cell wall as well as the central vacuole and the nuclei present towards the periphery, because the cytoplasm will be pushed towards the periphery. So let us discuss the features of collenchyma tissue first. features of collenchyma tissue. So again, these are also living cells, polygonal shaped cells, but they are elongated cells. So when we say polygonal and elongated cells, they have the central vacuole, cytoplasm, and the nucleus is present. And the most important and distinct feature, corners of the cell wall are thickened with cellulose. Here also you have to remember, students, still the cell wall is cellulose, but the corners are thick. Because the corners are thick, intercellular space may be absent or not visible. So using these features, you should be able to identify the collenchyma tissue from a plant tissue. Now that you have understood the features of collenchyma, we will observe this in the lab using permanent slides. Now we are going to look at the second type of simple permanent tissue, that is the collenchyma tissue. We have already discussed the features and you know how this differs from the first type, parenchyma. So now I'll select the tissue that is relevant to this. Okay, this is the permanent slide that has the collenchyma tissue. So again, I will mount it on the stage. Now I make sure it's the low power lens and also there is enough light falling on the specimen. Now the slide is ready for observation. I will observe it under low power. Here students, you can observe that the cells are polygonal in shape. And also you can see that the corners of the cells are thick. You all know that it is cellulose. So these are the main observations that you can see from this slide. Now that we observed the collenchyma, you can remember the parenchyma tissue and you can now differentiate between the two tissues. You all observed the collenchyma tissue, you would have been able to identify the different features of collenchyma. Now that you have understood the features, now let us discuss the locations of collenchyma in the plant. Locations of collenchyma. The collenchyma forms a cylindrical tissue inner to the epidermis of herbaceous stems. So, a cylindrical tissue inner to the epidermis in herbaceous stems. You all know what herbs are or herbaceous plants are. The plants that do not have a woody stem, which are very short in height. So, these are the herbs. In that, if we take the cross section of a plant stem, it will be something similar to this. You know, this is the epidermis. The outermost layer is the epidermis. And below that, you will have a layer of collenchyma. That is one place where you find collenchyma in plants. That is especially in herbaceous stems. And also they are found in the veins of dicot leaves. You can remember, students, you have learned this in the smaller grades. The dicot leaf has reticular venation. Here all the veins are like a network. And in these veins there is collenchyma. And these collenchyma provide support to the plant leaf. Collenchyma in veins of leaf. So these are the two locations where you find collenchyma in the plant. Let's discuss the functions of collenchyma. Functions of collenchyma: support. Collenchyma provides mechanical support to dicot plant stem before the formation of wood. We have discussed this before also. In dicot stems, the diameter of the stem can increase. That is due to the cambium present, and this is known as the secondary growth, where the wood is formed. So before the wood is formed, the collenchyma that is present in the young stem provides the mechanical support. This tissue provides support to the plant leaves by the collenchyma in veins. So here also it is the collenchyma that is present in veins. So mainly the function is support. In addition to that, photosynthesis is carried out by collenchyma tissue. Chloroplasts are found in the collenchyma of immature dicot stems. You all know, students, young plants have a green color stem. That is the immature stem, and photosynthesis is carried out by those cells. So I am sure you understand the functions of collenchyma. Now let us move on to the third type of simple permanent tissue present in plants, that is the Sclerenchyma tissue. Sclerenchyma. Sclerenchyma tissue helps in providing mechanical strength and support to the plant body. So this is also similar to the collenchyma tissue. And these two differ from parenchyma in their function. This tissue has two types of cells. One is known as the Sclerenchyma fiber. Sclerenchyma fibers are shown here, or we can straight away call them as fibers. And the next type is sclereids. Sclereids. So here both the tissues are shown to you. This is a cross section of a fiber, that is a cross section of a sclereid. Here you can see the fibers are usually elongated cells. If you look at the longitudinal section, they are elongated long cells. And if you look at the cross section, they are polygonal in shape. And in the cell wall is made up of cellulose, there is a substance called lignin. The lignin deposits in the cell walls of these tissue. So because of that, the cells die. So the sclerenchyma tissue is made up of dead cells. Because the cells are dead, the cytoplasm, central vacuole and the rest of the organelles are not present in the cell. And therefore, the center part of the cell becomes the lumen. This is called the lumen. So students, in this diagram, the cross section of the fibers, here you can see these connections. And here that is very much visible. There are connections between the adjacent cells. Those are known as, in this longitudinal section also, it is even more clear. Here you can see those connections and those are called the pits. They provide connection between the two adjacent cells. So these are the features of sclerenchyma tissue. The fibers which are longer and the sclerids which are short cells. So if you are given the diagrams, you should be able to identify these tissues. And here you can say fiber is written as FIBRE or it can be written as FIBERS. Both refer to the same thing. But when we refer to sclerenchyma fiber, we have to say sclerenchyma fiber or sclerid. And you will see when it is present in different tissues. Like xylem, we call it the xylem fiber. And in phloem, we call it the phloem fibers. So now that you know an introduction to sclerenchyma, I will move on to the features of sclerenchyma tissue. Features of sclerenchyma tissue. So I explained that the cell walls have lignin deposit on them. So because of that, the cells are dead. So if we write down the features of sclerenchyma, there is lignin deposit in the cell wall. Therefore, cells are dead. And with this lignin deposit, because the cells are dead, all the components of the cell or the structures of the cell are not present in the cell. So there is no cytoplasm, no organelles. So here I can say there is no cytoplasm or organelles. Therefore, the space inside becomes the lumen. So the next feature is that a lumen is present. And around the lumen, if you look at the cell wall is evenly thickened. Cell walls are evenly thickened. And because of all these features, you saw that the cells were very tightly packed. So the cells are tightly packed. So students, the cells of sclerenchyma tissue, because of the lignin deposit or what we call as lignification, the cells are dead cells. there is no cytoplasm or organelles, the center part becomes the lumen and the cell walls are evenly thickened and the cells are tightly packed. So, those are the features of Sclerenchyma tissue. So, now we will observe a permanent slide of the Sclerenchyma tissue in the lab. Now, we will observe the third type of simple permanent tissue, that is the Sclerenchyma. So, for that I will first select the tissue, right, that has the Sclerenchyma tissue here. So, I will place the permanent slide and also I make sure that it is in the correct position and enough light is falling on it. And first we will observe it through the low power. Now, when I do the adjustment, coarse adjustment, you can see clearly the cells that are present in Sclerenchyma. You can see the evenly thickened cell walls and the lumen present inside. And also the shape is polygonal. There are polygonal shaped cells with the thick cell wall and the lumen present. That is what you can see through the low power. To observe it a little more clearly, we will now observe it through the high power. So, here I will use the high power to show you all the Sclerenchyma tissue. And when I do the fine adjustment, you can see it more clearly. The polygonal shape of the cell, the Sclerenchyma cells which have the thick lignin cell wall and the lumen in the middle. So, that is a distinct feature which you can use to differentiate Sclerenchyma from the other two types, that is Parenchyma and Collenchyma. So, with these students, we have observed all three simple permanent tissues and you will be able to differentiate and identify them separately. Okay, students. Now that you observed the Sclerenchyma tissue, you would have been able to identify different features. With that, we will move on to the locations of Sclerenchyma tissue. Locations of Sclerenchyma tissue. Fibers are present in xylem and phloem. Now, we have been talking about the vascular bundle. The vascular bundle is the transport tissue present in the stem, leaf and the root of plants. In that, there are two main types of tissues. We will be discussing them later, the xylem and the phloem. In both xylem and phloem, there are fibers. If it is present in the xylem, we call it the xylem fiber. And the fiber present in the phloem is called the phloem fiber. So, these are two types of fibers present in plants. Then there are other parts which contains fibers. You all know the coconut. Around the coconut there is the husk which contains coconut fibers. This is used to produce coir. So, that coconut fiber is Sclerenchyma fiber. Then agave fibers and cotton wool are all fibers. So, you have to remember these examples. Coconut fiber, agave fiber and cotton wool are made up of fibers, that is Sclerenchyma fibers. Then if we look at the sclerites, they are found in different parts of the plant. They are found in the endocarp of coconut, camphor and mango fruits. I explain what seeds are In this, we discuss different parts of a seed where we get parenchyma tissue. If you take the coconut, you have the shell of the coconut. Inside that, the white color part, which is the food storage that is used for cooking, is the endocarp. So this endocarp is crunchy. If you take a piece of that white color endocarp, that is edible. And if you bite it, you eat it, you know it's crunchy. The crunchiness is because of the sclerids present. Same goes for camphor and mango fruits, mainly in the raw state. Then we have the pericarp of guava fruit and in pear fruit. Endocarp is inside, pericarp is outside. So, around the seed, if you have the fleshy part, that is known as the pericarp. Here, this is inside the shell. So, when the pericarp is crunchy, like in the guava fruit and pear fruit, you know it's due to the sclerids present. Then we have the seed coat of coffee and dates. Those are also very thick, strong seed coats. So, all these contain sclerids. And as you can see, not only the structure of the sclerenchyma fiber and the sclerids are different, but also the locations of these two types of sclerenchyma tissue are different. Now that you know the locations, we will discuss the functions of sclerenchyma tissue. Functions of sclerenchyma, there's only one function. They are all dead cells, and because of that, they only provide support to the plant body. So, the lignin that is present in the cell wall helps these cells to provide the support. So, students, with this, we have come to the end of simple permanent tissues. Next, we will discuss the complex permanent tissues present in plants. Complex permanent tissues. As we discussed before, different types of cells together form a complex permanent tissue. If it is only one type, it's a simple permanent tissue. If there are more than one type of cells, it's the complex permanent tissue. So, different types of cells. Complex permanent tissues are found in vascular systems of root, stem and leaves of plant. So, the vascular system is the system that is there in the plant for transport of substances. It transports water and minerals together, and also the food is transported. When we transport food, we call it the translocation of food. So, the vascular system transports water and minerals and translocates food. And based on the function, whether it transports water or translocates food, there are two types of tissues. One is the xylem tissue. And the other one is the phloem tissue. So, based on their functions, there are two types, the xylem tissue and the phloem tissue. Given in this diagram is part of a cross-section of a stem. We did observe this before. In this, you can see this part is the vascular bundle. And in the middle, we talked about this part, that is the cambium, lateral meristem. And on either side, you get the xylem and the phloem. And normally, in a vascular bundle, the phloem is towards the outer side of the stem, towards the epidermis of the stem. And the xylem is inside. So, this part is the xylem and this dark region is the phloem. Both of them together is known as the vascular bundle. Now we will discuss the xylem tissue in more detail. Xylem tissue. Two diagrams are given to you. Here you can see these are more tube-like structures. But if you look at the tubular structure in more detail, these tubes are made up of small cells. The cells are all joined together to form the tubular structure. So that tubular structure is known as the xylem vessel. You can see that here as well. This is a xylem vessel and these are the cells that make up the xylem vessel, and we call those cells as the vessel elements. So there is one type of cell present in xylem tissue, that is the xylem vessel element. Then similar to xylem vessel elements that are there to transport water, there are these tracheids. Tracheids are long, elongated, spindle-shaped cells. So that is the second type of cell present here, tracheids. In addition to these vessels and tracheids that mainly transport water, there are xylem parenchyma and xylem fiber. Xylem parenchyma is there to store food, and also xylem fiber is there as a mechanical support tissue. So the other types of cells here are xylem parenchyma and xylem fiber. So as you can see, students, there are 4 different types of cells. So that confirms this is a complex permanent tissue. Now let us discuss in more detail the structure of xylem. The xylem vessel elements are cylindrical, elongated cells. You saw in the diagram, they were like cylindrical in structure and long cells. And these xylem vessel elements arrange one on top of each other, and the cross walls, the walls that are there as the cross walls between the two elements, dissolve. And therefore, there will be a continuous tube-like structure. That is the xylem vessel. Xylem vessel element stack on top of the others and the cross walls are dissolved. So here you have to remember this term, cross walls. They are dissolved to form a continuous xylem vessel. So the xylem vessel elements are individual cells, but the xylem vessel is a continuous tube. This tubular structure helps in transportation of water in plants. So this is the main structure that transports water in large plants. Then we have the tracheids. Tracheids are elongated, spindle-shaped cells. You saw the shape in the diagram. They also help in transportation of water. There are some herbaceous plants that only have the tracheids for transport of water. But the main element is the xylem vessel that transports water. Then we have xylem fibers. Xylem fibers are narrower and shorter than tracheids. They provide support to xylem tissue. So as we already discussed, Sclerenchyma, the function is support. So the xylem fiber also provides support. Xylem parenchyma. Xylem parenchyma are living cells with a thin cell wall. They involve in food storage. So here students, you have to remember the xylem vessel and the tracheids are going to transport water from the root to the top of the plant, through the stems to the branches, the leaves. So the water has to be transported throughout the plant. And you have seen very tall plants. There, the water has to be transported against the gravitational pull. So because of that, water has to be transported with a very high pressure. In order to withstand this high pressure, the xylem vessel as well as the tracheids and the fibers present have to be very strong. There should be a high mechanical strength to these cells. In order to provide this high mechanical strength, all these three cells are lignified. There is a lignin deposit in the xylem vessel element, tracheid and the xylem fiber, the cell walls. So because of that, all these three cells are dead cells. So you have to remember students, there is lignification. Lignification of cell wall to provide strength. So this makes the xylem vessel element, tracheid and the xylem fibers to be dead cells. So the three types of dead cells, the xylem vessel, tracheid, xylem fibers. In this, there are three types of dead cells and only one type of living cell. That is the xylem parenchyma. So as you can see students, xylem is a complex permanent tissue because it has more than two types of cells. There are four types of cells. Out of those four types of cells, xylem vessel, tracheids, xylem fibers and xylem parenchyma are all the four tissues. And the first three are dead cells and only the xylem parenchyma is a living cell. So now that you know how the xylem tissue is formed and the types of cells present in it, let us observe a permanent slide of xylem tissue in the lab. Now we are going to observe the two types of complex permanent tissues. So first, I will show you all a permanent slide of the xylem tissue. So here I have the xylem tissue, a permanent slide. I will place it on the stage and get it ready to observe. Right. So the specimen is in place and there is enough light. We can observe the tissue. You know that xylem tissue is made up of different types, 4 different types of cells. So when you observe the tissue, try to identify the different types, especially the 4 different types of cells present in it. Now, when I do the adjustment, to a certain extent, you can see that there are different types of cells present in this slide. In the xylem tissue, there are different types of cells. You can observe the different shape and the sizes. So, from this observation, you will be able to identify the xylem tissue as a complex permanent tissue. Now that you have observed the xylem tissues, you would have been able to differentiate the different types of cells present in it. Now, let us move on to the functions of xylem. Functions of xylem. Transportation of water and minerals to the plant body which are absorbed by plant roots. So, here transportation of water and minerals. You have to remember that, students. And also you have to remember in very tall plants, because of this transport takes place with high pressure, the cells of the xylem tissue are lignified. There is lignin deposit in the cell wall and they are dead cells, so that they have a high mechanical strength in order to withstand the pressure exerted by water, providing mechanical support to plant body. So, as I mentioned, because they are all lignified cells, they themselves can provide support to the plant body. So, those are the functions of xylem tissue. Now, I will move on to the second type of complex permanent tissue, that is the phloem. Phloem tissue. This is also a complex permanent tissue. Therefore, it is made up of more than one type of cells. First, we will try to identify the different cells in the diagram. Here you can see a longitudinal section of the phloem tissue. This has sieve tube element. Here you can see sieve tube element. That is the main type of tissue that translocates food. So, here again these are long elongated cells which are connected together, but the important thing is the cross walls dissolve incompletely. So, because of that, at the cross walls you can see sieve plates and pores there. You all know what a sieve is. We sieve different solid substances. It has pores in it. So, similar to that, this sieve plate also has pores through which the food can be translocated. So, you can see the sieve tube element and the sieve plate. Because food has to be translocated through this, the cytoplasm, nucleus and the organelles of the sieve tube element are disappear. And because of that, the sieve tube element cannot function on its own, but still it is a living cell or it is a living tissue. To control the activities of the sieve tube element, there is a companion cell with a prominent nucleus. And the nucleus that is present in the companion cell controls the function of the sieve tube element. So, we have discussed two types of cells that are present in phloem tissue. One is the sieve tube element. These are living cells, but they don't have the cytoplasm and the organelles, but they have a space in between so that food can be translocated easily. And the companion cell that is adjacent to the sieve tube element, and this controls the activity of the sieve tube element, and the distinct feature is the sieve plate. The same things can be observed in the cross section. Here you can see the sieve plate with the pores in it and the sieve tube element, and also the companion cell with a distinct nucleus. In addition to these two, there is the phloem parenchyma. Again, that acts as the food storage cell. So, we have phloem parenchyma. And also to provide support, there is phloem fiber. Phloem fiber. So again, you can see there are 4 different types of cells present in the phloem tissue. Therefore, it is the complex permanent tissue present in plants. Now, let us discuss the features of phloem tissue in more detail. Sieve tube. The sieve tube elements fuse end to end and the cross walls are incompletely dissolved. I discussed that with you all to form a sieve tube. The cross walls in these sieve tubes are called the sieve plates with the pores in it. Sieve tubes transport food, mainly sucrose, throughout the plant as a solution. If you can remember, students, when we discussed the first lesson of grade 10, we discussed carbohydrates, and in that we saw sucrose is the substance or the carbohydrate in which form food is translocated throughout the plant, and there we call it the cell sap or phloem sap. Phloem sap contains sucrose. So here if I write that name, this is known as phloem sap. That is the sucrose being translocated through the phloem. Then we have the companion cells. Companion cells are elongated cells associated with sieve tube elements. Because the sieve tube elements don't have a nucleus, the nucleus of the companion cell controls the activities of the sieve tube element. So, this is something that you have to remember. Sieve tube elements lack a nucleus. Then we have the phloem parenchyma for food storage and phloem fibers that are found scattered in the phloem tissue. So, like we discussed before, there are dead cells and living cells present in the phloem tissue as well. Out of the 4 types of cells, 3 of them are living cells. So, if I write down the living cells, the sieve tube element, the companion cell, and the phloem parenchyma. These 3 are living cells. So, that means they all have cellulose cell wall, and there is only one type of dead cells, that is the phloem fiber, and you know the reason why. That is because there is lignin deposit in the cell wall of fibers. That is a sclerenchyma tissue. So only type of dead cells are the phloem fibers. So 4 types of cells: sieve tube element, companion cell, phloem parenchyma. They are all living cells and only the phloem fiber is a dead cell. This is different from the other type of complex permanent tissue that we have discussed before, xylem. Xylem has 3 types of dead cells and only 1 living cell. But here there are 3 types of living cells and only 1 type of dead cells. So students, now that you have understood the structure of phloem, we will observe a permanent slide available in the lab. Now we are going to observe the second type of complex permanent tissue, that is the phloem. So in the phloem tissue as well, there are 4 different types of cells. And the most distinct feature is the sieve plate. You can see the polygonal shape and the dots, that is the pores in the cross wall. So from that you can easily identify the tissue as the phloem tissue. So for that again I will get the cell, the slide that has the phloem tissue. This is the permanent slide. I will set it to the correct position. Yes, now there is enough light falling on it. Okay, so we can observe it under low power. And the lens is also low power. So if I look at this tissue, you can see, when I move it around, you can see the sieve plate very clearly. Can you all observe the sieve plate, students? There are 4 different types of cells, but out of those, you can clearly see the sieve plate. So that means this is the phloem tissue. So with this observation and the previous observations, we have observed the different types of simple permanent tissues and the complex permanent tissues. So when you are given a diagram or if you are given a slide to observe and identify, you know how to do that. You have to look for the distinct feature. Okay students, so you would have observed the different types of cells present in the phloem tissue. We will move on to the functions of the phloem tissue. Functions of phloem tissue. The food synthesized in the leaves are transported throughout the plant body by this tissue. We have discussed that function before, and the process is known as translocation. So you can remember students, glucose is the main product of photosynthesis. It temporarily stores as starch, then it is converted to sucrose and the sucrose is translocated by the phloem to the lower parts of the plant. And then we call that substance as the phloem sap. Okay students, with this I have come to the end of discussion on plant tissues. I hope you have understood all the different types of tissues, simple permanent tissue and complex permanent tissue, and also the other main type which divides all the time, the meristematic tissues. You have to understand the features, the locations where you find these tissues and the functions of all the tissues. Now let us summarize the different types of tissues present in plants. So this is a diagrammatic form of the classification of plant tissues. How do we classify them initially? As meristematic tissues and as permanent tissue. Then we classified the meristematic tissue based on the location present. So we had the three types: apical meristem, intercalary meristem, and the lateral meristem. Then we have the permanent tissues which have differentiated cells, and in that, if there is one type of cell only, we call it the simple permanent tissue. So these are the simple permanent tissue. What are the three types of simple permanent tissue? Parenchyma, Collenchyma, and Sclerenchyma. And then we have the second type of permanent tissue, the complex permanent tissue, which has more than one type of cells. So here we have the complex permanent tissue with more than one type of cell, and they are usually the vascular tissue. The two types are xylem and phloem. So students, you know the features and the locations and the function of all these different types of tissues. If you are given a diagram, you should be able to identify the different types of tissues. With that, we have completed the discussion on plant tissues. Next, we will move on to the second topic, animal tissues.